zoom out again now? You just scoot over a little bit.
How's my audio now?
Oh, keep speaking with another full sentence.
Okay. I will, because Erica's providing us with these wonderful facts about the octopus. So one thing is that this octopus does eat small crustaceans, but also fishes, and is eaten by fish. And then the other thing to know is their eggs may take up to 450 days to hatch. So octopus, when they lay eggs, they sit on them and they don't eat. The octomamas don't eat the whole time. So that means that an octomom, after laying these eggs, for much longer than a year, would be sitting in one place protecting them.
Oh, that octopus is right in the way right now.
Yeah, we better
I
get
know, I
the
wonder if
sample
octopus eats
before.
small brachiopods.
Yeah.
Okay, can we zoom in now? Yeah. Okay, move on, Mr. Octopus. Yeah, that looks like a grabbable. Oh,
Oh,
Oh, wow.
oh, it's swimming away.
wow.
Oh.
Oh, wow.
Okay.
That makes the collection even harder.
Oh, it's coming back.
That was a surprise. You okay? You get a little shock over there? How often does that happen?
That reminds me more of a scallop,
Surprise.
the way it
Yeah.
swam.
Yeah,
It looks so attached.
yeah. Huh.
I lost it, but I think it's somewhere under your porch.
Yeah.
You zoom-
Are we
Yeah,
chasing after it?
What do you want to do? Sure.
Can you zoom in a little bit?
On what?
Let me move back
Yeah.
a bit.
It
Okay, can you
definitely,
zoom in now?
now it's
I missed what we were shooting.
right by the laser. It's just below the laser right now, I
I
think, isn't
think
it?
so, right? Is
Yeah, I
it,
can see it.
Rock?
It's not the lightest one. It's slightly
Going
Yeah,
in.
kind
there
of
it is.
camouflaged
Yeah.
on top of the rock.
You think that's
Wow.
the same one?
Yeah.
Yeah.
Yeah. That's
Try
it.
again. Yeah, I see it.
Well, I mean, this is definitely a slurp thing. We got to sneak up on it.
Yeah. So yeah, Daniel is saying it's a pectin clam. Oh, sorry, pectided clam. Which pectin, the root of the word, equals scallop. Interesting.
So
There's a small little branching
are we after it?
coral
Then?
maybe underneath also.
I'm asking.
I gotta rotate a bit to get more zoomage.
He said, will be very difficult to collect, and very was in all caps.
I think we tired it out. I think if we sneak up behind it.
Yeah. Yeah.
The idea of trying to sneak with an ROV is kind of silly, but
But we're willing to try.
yeah, we might get away with it.
Oh, he doesn't have eyes or ears. No?
I wonder if these are the scallops that have the light sensors. Some scallops have
Yeah.
what do they
He's getting enough light, so.
yeah,
Ready for
Okay.
zoom?
probably not at this
Do we
depth.
know we're? Well, I mean, from us, there's definitely no light at 1,500 meters.
No, not for these.
All right, flushing now. Which jars are clear?
Any of them. They're all open. First sample of the next cruise.
Okay. Ready
the
for
first dive.
suction.
Yeah, we get lined up here. Okay, you got
We have
suction?
about
Turning it on now. All right.
And you see, he's gonna swim towards me, right? Because he's gonna shoot. He's gonna propel himself towards me.
If you put the hose right on top of them and just crank it to 100, no?
Yeah, well, I think we'll crank it and then we're gonna stick it on there. Okay. Here we go.
Very, very quiet. Oh,
Got him.
Oh, nice.
oh my gosh. Such drama.
Steve, when you have a second, what sample is this going to be?
Yeah, this is going to be number 1001.
Great, thank you.
Still don't see him in the jar.
Wasn't that big. He should go right in.
Boil this again.
Maybe it's swimming its way back out.
Yeah. Well, I've still got the suction on.
We only have about 120 more seconds for public questions, and I do want to ask one more. You don't have to answer it now, but the question is, are you ever worried you might be going past new species of tiny animals, like the things you can't see on the rocks or floating in the water?
I turn
That's
it
going to
off,
be
see
probably
if,
our last question.
It drops down.
Yeah.
Thanks, Claire. Yeah, so if the public
Yep.
notices sometimes
Yeah.
when we sit down and zoom in,
I
you can
think
see
that
a
was
lot
only half
more
one.
the smaller community, smaller benthic community that we don't normally see when we're doing a transect or flying, a meter or two
There it is.
above the seafloor. So it kind of depends on the science objectives for the dive. But when we do stop and we find some interesting things and our scientists ashore who are joining us say they want it, or they want us to zoom in or sample something, we try to accommodate those requests.
Okay.
And to confirm, that was jar number 7.
7?
Yeah.
Got it.
And then Liz Clark is also curious about that
Look
little
at that. I
sponge
did
that
that
I
with
think
an
is.
arm and like.
Jess mode.
Yeah. Wow.
Kurt, could you tell us where that you see the sponge right now?
I believe it's the one that is directly in front. It's to the left of the laser is about 8 o'clock, maybe.
Oh, to the right of
Nope,
the
not
octopus?
to-
Oh, no, that's a shell. To the bottom
no, I
left
think it
now?
was
now?
really. Let's see if. Let's see if.
What are you looking for?
A yellow
I think
sponge.
it was to the right
Oh.
a little bit. It's right next to a brittle star, and then maybe a little
right.
comment it.
Right there.
How about a white sponge?
Yes, that's the one. I believe she's asking. Let me just see if she confirms.
Zoom in. I'll try and get rid of the shakes here. No can do. Zoom out now.
Yep, that was the one that you had zoomed in on. She's interested in slurping.
Okay. Let me get off this rock.
Sure.
Can you zoom in again now? What's that on top of the rock? There's another octopus even smaller.
Whoa.
Oh my gosh.
Oh, wow.
Hello.
That's really dinky. Wow.
If they get any smaller, we're going to need that extra zoom.
No zoom for you. Bob, can we do a DVL reset here, Bob?
Yeah, go for it.
Okay.
So I tried to restart my computer. Has that improved my audio at all?
Right now it
Sound
sounds
pretty
like
good
it.
to me, Jen.
What's that?
Sounds good so far.
Okay. Well, I can hope.
Oh, that's
Yeah.
definitely, that's better.
Yeah.
Yeah.
Okay. Okay.
Yep.
I hope
Yep.
that's it.
Good.
I missed
We'll
a
keep our
little of
fingers
the
crossed, Jen.
excitement. But I think I see the octopus that you're talking about. I'm still getting everything expanded. But coming in from the right,
Yeah.
that's what you're looking at?
Yeah.
I haven't seen it compared to the lasers, but I think there was a
It's like
request for
2
us
centimeters
to,
or something long.
oh,
You
wow,
want to zoom out
adorable.
again now?
To mention that those lasers
I
are
think we
10
have Marley now.
centimeters
Zoom out.
across.
Oh. There, well, not that much. Just so you get the, just frame with the lasers and the octopus.
Oh, gotcha. Yeah, thanks.
So if you're looking at those lasers are 10 centimeters, and you can see that the octopus is only probably, like Bob said, about 2 centimeters.
Yeah. It's pretty dinky. Okay.
So as the voice of the public, I'm signing off here, and I hope people continue watching, but we are disabling comments for at least a few hours. So goodbye and good luck.
Thanks, Claire.
you, Claire.
Appreciate
And
it.
sorry, everyone, we couldn't get to all the questions. I'm glad to hear that there's so much interest, and we'll try to keep thinking what it is you might want to know and offering it up to the best of our abilities.
So are we interested in zooming more on this octopus, or are we going over to look at the sponge again?
I think we can go to the sponge, Bob.
Right? You zoom in some more?
All right.
Oops. All this stuff's pretty small. Okay.
Yeah, and if it makes sense, and maybe Steve has some thoughts, but we could slurp this in the same bottle as the scallop, unless Daniel chimes in to say definitely a no-no. But
It's fine with me. We can do that.
All right.
Probably won't need as much force
I don't need
for the
this.
sponge.
What are you doing there? Need that. Need this. Okay, can we zoom back out?
yep, and Daniel confirmed same bottle is fine.
Yep. So that's going to be jar 7.
Yep.
For both.
Okay.
Yeah, if we think the sponge is going to be easier to slurp, I'll use less suction.
Oh.
Here.
Okay, can we zoom back in?
All right.
Got it on?
Ready?
Yeah. We probably should have just picked up the rock, because it's... Is that too messed up? Would you rather pick up the rock? It's not very big.
Sure.
Yeah? Okay, zoom out.
Yeah, that works.
Look out, starfish.
Going fast.
Wow.
Missing a leg too, or an arm.
Yeah, sorry
Yeah.
about that.
Oh. Okay, they grow back.
Thanks for pointing that out.
Sorry,
Yeah, it wasn't-
I didn't know. Brittle spurs are very fragile, but they grow back very quickly.
You can throw this in one of the forward box compartments.
Okay. You want to zoom in some more on the sponge? Got a little divot in it.
Makes it easier to get off the rock later.
Yeah. Okay. Zoom out. Box out.
All right, box coming out.
Which side? Which side are we going in? This one?
Yeah, anyone.
Oh, it doesn't matter. Okay.
Great.
Press my luck there.
I was gonna say bold.
That's great. Thanks. All right. I think we can, whenever you are all ready, you can probably keep heading in the same direction.
Sounds like a plan.
Okay.
Steve, that was sample 002, correct?
Correct.
And what box is that?
That's in forward A.
Thank you.
I'll let you guys catch up to the ship before putting in a move.
Okay. Taking off.
Bye, octopus.
Same.
Bye, octopuses.
Or octopi?
Octopi is,
Oh.
I've heard recently, is not correct.
It's not correct.
I believe
That's a singular.
I believe octopus can actually be used for singular or plural, or octopuses.
Is
First
it
side, is
similar,
that-
Sam, to, I was going to ask you, because I hadn't heard that, but it makes sense given the convention for fish and fishes. But
Yeah.
Fishes is usually talking about multiple species of fish, as opposed to multiple individuals of the same species.
That's what I've heard too. We can go for that. For now.
Sounds confusing.
Just stick with octopus. Can't go wrong.
Alright.
Or if you want to get really technical, octopods
Wow.
There you go.
or octopodes.
All right, I think we can call on that ship move.
Yeah.
Let's do it. Bridge nav.
Are we just cruising, or is this another transect?
I think we're just cruising.
30 meters, 135.
So we had a
Can
request
we,
for the name
zoom
on
in
that
again?
white cucumber that we've been passing over. And at least one of them.
Anemone.
Well, I'll wait till we see a new one, and
Anemone.
another one, and I'll point it out.
Okay, zoom out.
Looks like some kind of anemone.
Yeah.
We never did
Maybe
intros for this
down in the
watch.
sediments. Oh. Good point, Al. I think you want to start, don't you?
Me and my
I love
big
it.
girl. Al Santos, video engineer for the 8 to 12 watch.
Hi, Paul Glick, the Argus pilot.
Bob Waters, a Herc pilot.
Samantha Wishnack, navigator.
And the data logger's chair, Steve Oskovich.
All right. And I'm Jennifer Brown, and I am one of two scientists that's talking to you from our home offices on shore. And sorry to folks, I heard that the audio was a bit annoying. I hope I fixed it on my end. And I'm excited to get a chance to explore the Santa Lucia Bank here. And I typically am out with the Nautilus looking at parts of Monterey Bay National Marine Sanctuary or Channel Islands National Marine Sanctuary. So this is a new area
Are you
for me.
interested, sorry to interrupt you, are you interested in a benthic siphonophore
No,
to the
go ahead.
left? See that thing bobbing? The little
In
pom-pom?
looking at it?
Yeah.
Yeah, let's look at it.
Yeah, for sure.
To the left lasers. Yep. Looks like a little.
And that was
Sorry, Bob.
Thought we were really getting
Words
excited
weren't
about the
working.
Scarlet King crab over there.
We zoom in out. Ow. Ow.
Troubleshooting going on. Oh yeah.
Whoa.
Nice.
That's
little
jelly. What is
Very
that?
cool.
benthic siphonophore, or is it a benthic ctenophore?
No, it's a siphonophore.
Siphonophore, yeah.
Really?
So you can see the little lines that it's attaching to the seafloor with.
Like fishing
Yeah,
line
it's-
coming off it.
Off to the left, yeah.
And the right also.
And the
I see
right,
them.
yeah.
Is that all the way in?
That's all the way in.
All the zoom.
Sorry.
Alright.
Well, I need another lens.
These creatures are too
Yeah, so
small.
Wow.
Erica Burton from the Monterey Bay Sanctuary gave us the name. It's Dromalia alexandri, and Erica Frew at the Northwest Fisheries Science Center reminds me they're affectionately, I think, called sea pineapples.
Oh.
Perfect.
So these are colonial members of the same group as the jellies and the corals and stuff like that. And Steve, you are so knowledgeable on the corals and the anemones and stuff. Are you also pretty knowledgeable on these?
No.
Benthic siphonophores?
No, my only experience with these is my first cruise out on Nautilus. We slurped one, and I was up for most of the night trying to pick out all the dissociated little zooids out of a slurp jar.
Oh.
You don't want to
Because it is
slurp
colonial and
they totally just like dissociate once you get them in the jar. They disintegrate.
Sand, don't slurp it.
Yeah. I think since we have a good species ID, I think we can
move on.
It's a really
Quickly.
polite scientific way of saying,
quickly. It's
please
bringing
don't slurp.
flashbacks. Yeah.
Is that a little cup coral?
Okay.
This other thing? Can you zoom in again? Little tiny thing there.
It's like an anemone.
Oh,
Yeah.
is that type of,
All right.
some type of worm up above it, too?
Oh, maybe that's what it is. It's just a plume sticking out. All right.
Yeah.
You good? No.
Yes. Thank you. And Sam, how far are we away from the end of the last transect we did? Transect 1?
We are,
You didn't move very far.
yeah, we didn't move very far. We're about 50 meters.
I didn't think so. I just
Yeah,
50
Yeah.
meters, 50?
50.
Okay.
What are we looking for now? Are we still looking for brachiopods?
Well, I think we're looking for more rock that has corals and sponges primarily, but we'll keep our eye out for brachiopods on the way. Does that sound good, Kurt?
Yeah, that sounds good. And then if Daniel chimes in that he sees something really interesting, we're keeping an eye on the chat here.
Okay, sounds good. We didn't get a chance to hear your introduction, Kurt. We might have a
Yeah,
moment
thanks.
here between cool stuff.
Sure. Yeah, my name is Kurt Whitmire, and I work at the Northwest Fisheries Science Center. So I actually work with some of the other watch leads that you all have heard on previous watches. So Erica Frew, Abby Powell, Meredith Everett, Liz Clark, and then some of our counterparts at the Southwest Fisheries Science Center with Tom Leidig and Di Waters. And I'm coming to you from Monterey, California, which is my home. And yes, I believe Sam's birthplace.
Yep, and current home.
And current home, good to know. How come you never come and say hello?
Oh,
Because you're always
There we
out there, right?
yeah, always out here. And actually, Claire is also in Monterey, but she's
Oh, nice.
not on the call anymore.
Okay.
What is that interesting shape that the laser just passed over?
The flattened sea cucumber?
What was that? Yeah, I was thinking cucumber.
Yeah,
It's a little
I wonder if it's
depressed.
a pyrosome, a dead pyrosome.
Oh, that seems likely.
Oh,
What? lots of sediment on it. Wow.
We did see a lot of pyrosomes on the way down through the water column.
Okay, zoom out.
So while Kurt was introducing himself, you may have noticed we passed over a rock that had a sea cucumber under it, and next to that there was looked like a tunicate, and then a large sea star. In case anyone was wondering what those things were.
And Steve, Erica Burton commented on that appendix I found it for. She had a species name correction for you.
I got it. I corrected it in the log.
Okay, thanks.
Thanks, Erica. I've been consulting one of at least the references for looking up some of these animals is the taxonomic guide to Sir Rich, which is a rocky structure in Monterey Bay National Marine Sanctuary that has similar depths to where we are right now, and it's a great resource for identifying animals at this depth. What is the white?
Yeah.
Ball? Is that a sponge?
Spongy.
Could be spongy, right?
Another dead pyrosome
I'm
there
not
under
sure.
the rock.
Yep.
It's very shiny looking.
How about top left, little orange specks? Is that spongy?
Left.
I don't quite see you.
Oh, now bottom
Abby's,
Oh, really?
right. Really tiny.
Man,
Sorry.
should have brought the microscope.
I know.
Oh.
so,
Erica
Al,
first
you're going to
noticed,
love this.
Mhm.
Oh, it could be eggy. They're asking
Eggs,
for more
yeah.
zoom. I don't know if we have more.
There are a couple there
Hygiene.
underneath the brittle star.
Oh, yeah, there you go. I see.
A little in the
Oh,
foreground.
wow. Did I?
It's
Is that any better, Al? The ones that are underneath the brittle star?
yep, I
Wow.
get it. Thank you. Right of frame is the only brittle star I see. Ooh,
Little tiny chitin almost there, too. It's amazing when you zoom in. It's a great zoom.
It's kind of fleshy.
thank you.
Yeah.
Everyone in the van's unhappy with it.
Looks like eggs, Abby's. They're thinking
Yeah.
it's eggs, the white mass. So I
Interesting.
think we can
Okay,
rule that out as spongy and move along.
moving on.
But
Okay,
there is
Al.
a clavularia up on the right,
Right
upper
up there.
right.
Can we focus on the... Well, it's okay.
Too much
Can
to
we
look at.
focus on this
Steve.
thing right there? I know we
Yeah.
can't zoom on it, but can we focus on it? We got any...
Second,
I
better
can move up some.
see your telly. Okay, where do you want to focus?
Or maybe a Garcinia.
Yeah, it's a little octocoral, I think.
You want to try and get closer to it?
No, that's all right. Just wanted to see if it was a small stony coral, if it was a octocoral.
Full lens.
Okay,
All right,
zoom out.
satisfied. And
How would you know it's an octocoral, Steve? Would you be counting the tentacles?
yeah, you
No.
could make
Can you
out the
zoom in again?
tentacles.
Sorry.
Yeah, it looked like a
Mhm.
small soft
Okay.
coral of some type.
And you'd be looking for 8 of them, correct? The octocorals all have 8.
What am I focusing on, Bob?
Oh,
2 sticks? Something?
yeah. Are those eggs too?
Ah,
Or
I don't.
Yeah, I don't know. It's obviously
Those are
sediment
worm
covered, but
tubes.
yeah, it looks like
Meredith,
yeah, it's a worm.
stick-
Is that what it is? Is that all the zoom? Oh, I definitely should have brought the microscope.
Oh.
I'm going to
But,
need a therapist after this watch.
We need to get into some more
So
larger fauna,
Meredith
otherwise we're
Everett-
gonna have...
Yeah. But
Okay.
Meredith said
Are
there
we
was
carrying on?
a stick coral looking thing on the
Yeah,
right
one-
side that we can
yeah.
look for as we zoom out.
as we zoom
Look
out,
to the
I'm
right
zooming,
for
Bob.
a
Coming out.
Yeah.
Oh, on the left.
Off in the distance. So we're going to be looking off in the distance.
Oh.
Wait, to the right for something that could be a small bamboo or a Besta pluma coral.
Okay, zoom in again.
Yeah, right where the lasers are.
Oh, I see it. It's above the sea cucumber. It's kind of white. There you go. You're finding it now.
Oh yeah, there it is. Small bamboo coral.
All kinds of goodies if you start looking.
Ship stop so we can stay as long as we want.
Let me ask her if she wants it, but I think she just pointed out.
Zoom out.
Meredith says we can keep going.
Okay.
Very interesting blocky habitats, though.
But the white cucumber that's going out of view to the left, I looked that one up, and that looks to me to be Late Magone, Laetmogone. Pretty one.
Yep, and that was what Meredith said that they had looked up for the last watch as well.
All right.
That's a good view of it. So I don't know a lot about holothurians or sea cucumbers, but from what I do know, they basically crawl around and eat the sediment, and they're digesting little critters that they're consuming.
Amphipod,
But they're
the thing off to the left there?
yeah.
Yeah,
It does
actually
look like
a good
it.
sized one.
Yeah.
We thought it'd be good for a quick peek. We're seeing a lot of them.
What are we peeking at? The amphipod? More zoom out?
You kidding?
All right. We're putting L to the limits here on zooms.
And I've got you sounding much better, too. All at once, I'm good.
Thank you, Al. You're a miracle worker.
Right.
For once.
Good capture, so I think we can move now.
Okay.
All right. Thank you.
Out.
Now I have to zoom out, too?
And that brittle star is regrowing an arm.
Let me
That was
zoom in.
fast.
Everything's going well.
What a great day.
If only we could do a transect at that zoom level.
If only.
So,
Boy, you'd get dizzy in a hurry.
Awful.
so Kurt, you and Eric are thinking that might be a different species of cucumber.
Oh, no, it's breaking up.
I don't know my sea cucumbers that well, but Erica, I defer to her. And Panickia was the other option that she thought it might be. So.
Okay.
But there may be, there may be multiple species we're seeing. It's kind of hard to tell. They're definitely, some are more kind of iridescent, I would say, than some of the other ones. So.
Mm-hmm.
Are we moving or we just...
I'll let you catch up to the ship.
I think we're pretty much.
Yeah, I'm pretty caught up. You can start moving.
Well, how long is our tether? 30?
Yeah.
Oh, so it's 20. Got it.
I'm almost like in the
Yeah.
middle.
Oh, yeah. Okay. Bridge, Neff? Step 30 meters, bearing 135.
Yeah, we haven't seen a lot of fish, at least since I started to watch. It's about tons of different invertebrates.
Wait, zoom in out.
Okay. A nice worm. It's got a duster worm.
Acorn
And is that
worm?
yeah, acorn worm, exactly.
Zoom out.
Couple of them. Do we know why they're
Oh,
called
there's a little
acorn worms?
octopus.
Oh.
Right in the
Oh,
bottom
yeah.
of the screen.
Another one?
Same kind, huh?
Oh, yeah.
Zoom in.
Did I zoom in? be right
And
there.
that's not an insignificant number of octopuses. We've seen quite a few. And how far we moved since our last transect? Maybe 40,
About
50
60,
meters. Yeah.
Oh,
60
yeah.
meters from the transect, but
Well, I'm excited to see what else we find around here.
40
There's
meters
so
from the
much
sample
when we
site.
look closely.
Wonder if this is some sort of nursery habitat for octopuses.
They're all little bitty guys.
Ruh-roh.
Yeah. We have to keep our eye out. And we did see some larger octopus on our way here that were holding onto the side of the rock. That as we were ending the watch before, but we didn't look closely at them to see whether or not, they were covering up any eggs or anything like that. But we can keep our eye out now.
I mean, if I was a baby octopus, this is where I'd live. Lots of nice little cracks and crevices.
To hide, yeah, definitely.
And plenty to eat.
Mm-hmm. And like you said,
So
we haven't
Sam,
seen too many fish, but we've also been moving kind of slowly, and so they may stay away when they see us and our lights.
So Sam, when we do get 100 meters away from the end of the last transect, let's set up to start another transect, and Meredith has requested a Niskin before we get going.
Sure thing. We're at
But right at the start.
Yeah, we're about 70 meters right now, but I'll let you know when we get to 100.
Great, thanks.
Zoom in out.
Beautiful feather star, also called crinoids. And a little bit of kelp. Do you see that to the upper left there? A little bit of drift kelp.
Yeah.
Zoom out.
Is that a sponge under the crinoid? Can't tell.
I thought it was a cucumber, maybe.
Yeah.
Oh yeah, there you go. Now I'm seeing it.
Okay. Put on. We moving?
We are moving.
Yeah.
7 meters left on the ship move.
Zoom in.
But I'll put in another one.
Is that a bigger one? Oh, it's a scallop? What is that?
Looks like it.
Yeah.
It's a scallop. Yep.
Is that, there's another little, no, that's a worm.
You sure? No. Oh, yeah.
Yeah. Okay, zoom out.
Just put in another ship move for 30 meters. And once we're done with that ship move, we'll be at 100 meters distance from our last transect.
And where was it that we wanted to take the Niskin?
Before we started the next transect, so
Got
when
it.
when we have our ship stop.
It's like another scallop. Yeah, zoom in.
Yeah, let's zoom in on those.
Those octopus hiding underneath the rock.
Yeah, what's
Another
on
one.
the left right there?
Oh no, that's a acorn worm.
Okay.
And Sam, I did look up the acorn worm, and I guess it's just, this name is based on the shape of the proboscis.
Looks like there's 2 scallops there. One up in the crack there.
Oh, yeah.
So this may also be a cesta, which is a file clam. We saw those on a previous dive. I'm trying to remember which one.
Yes, and thanks, Daniel, for helping us ID these.
All right, I
Hey,
got some
going out.
captures.
That's good, thank you.
Hold on that. And out.
I hear a little voice in the background. Is that a Zoom?
Yes, please.
Well, for those of you who are, oh,
Okay.
is that actually, sorry, but I looked up and boom, carnivorous sponge, right?
It's like the one
That
we
was my guess,
collected
yeah.
A
last night.
I
Pluma.
like it.
Do they retract into that?
Yes.
Stick?
Meredith says yes. Or no, Abby says yes. I think to collection.
Yep.
Oh, are you gonna
Yeah,
collect
they're
it?
pointing out it.
Mm-hmm.
Are we? Okay, I'll stop.
Can we? I know you put in a ship move. Do we
Yep.
have time to collect
We
this?
can stop.
Okay.
No problem.
Okay. Thank you.
Cool. Yes, thanks.
Great. So this is an Pluma, which is a kind of sponge. That's known as a predatory sponge. And my understanding is that what you see sticking out from the sides are their spicules, but they're used to actually capture prey instead of being a filter feeder to get its food. And is this something can we zoom out to see the size of it? Is this something we'd want to slurp?
That seems to work the best.
Okay.
We've had really good luck getting them in the slurp.
Okay.
I wanna move in.
Steve, I think what we saw float by earlier was an acorn worm.
The purple one?
Yellow.
That's what I was
That one just floated by and it looked just like what you had
Yeah,
circled before.
I was thinking, yeah, there might be a couple different types of acorn worms here.
Yeah.
I've only ever seen them on rocks. Never seen any swimming. Interesting.
All right, we're ready for jar number 6. Is that good?
Works for me.
Good catch, Ian, Bob.
Hey, zoom in.
Ready?
Yep. Come here.
So, one of our scientists ashore, Abigail Powell, is our sponge expert, and she says, to the best of her knowledge, this will be the first carnivorous sponge collected from this area called Santa Lucia Bank. So that's pretty exciting. And the carnivorous sponges, I think were only recently really started to be understood and collected, and they're still being worked on to figure out how many different species there are. This one we saw here had a single stalk. I've seen some that look like this, but then start to branch a little bit, as well
Alright,
as some that look more like
I'll turn off suction and see if it drops in.
a palm frond. So there's a few different kinds. So this sample will contribute to a better understanding of that group of sponges and their distribution
Still don't see it yet, so I'm gonna give it some more.
in this area.
Yep.
So right now we're looking at, if you look at channel 3 or the quad view of your Nautilus feed, we're looking at the jar, which is at the other end of that slurp tube, trying to confirm that the sponge made it all the way through the tube and is safely in the jar, where we'll keep it until it returns to the ship and then can be taken off of the ship and preserved for future study.
Oh, there it is.
Okay.
All right, great, thank you. When Abby's contributed that the first carnivorous sponges were discovered in the Mediterranean.
Another one over there. Zoom in. No. Okay, zoom out. Carrying on.
Carrying on.
All right, so one of the other things we're doing here in this section of the Santa Lucia Bank that we're referring to as the PALM, P-A-L-M, is quantifying, looking for corals and sponges and running quantitative transects where we proceed along the seafloor, capturing video for later counting and measuring organisms, and we're moving along here to get to where we can start a new one of those transects. And in the meantime, looking for neat animals and corals and sponges that we might need for our collection.
Zoom in out.
Tube worm.
Is that eggs again or
Oh,
sponge?
That one's a sponge. Some kind of plate sponge, maybe.
You got lasers off? Lasers on. Oops. We good?
Good.
Zooming out.
so science, we are just about 100 meters from our last end of transect. So anytime you'd like to start, we can stop and take a Niskin and then start a transect.
Great. And
We need to get caught
well,
up here.
and then Al, could you also put the high pack into channel 3? We have a request from some of our scientists ashore.
All right, back's up there now.
Thank you.
We're starting to transect again?
We are needing to stop at some point, take a Niskin, and start a transect. So Science, do you have any preference on where we stop to do this?
No, no preference. Yeah, as long as we're 100 meters away, we'll just stop and take that Niskin before we start our transect.
Alrighty, anytime then.
Yes, I can hear you.
This looks like a good place to stop, and we'll do a Niskin here.
Yep. Yes, I'm on NSPL as well.
Steve, this will be 004.
What Niskin are we doing?
You could start with number 1.
Oh, we haven't done any.
Or number 6. Whichever way you want to go, forward or back.
We'll go 1.
Yeah, I mean, I think we should be gently traversing upslope, but we're kind of trying to gradually move up to the top of this ridge.
Fire? See?
If Guy has a different preference.
I think I see one vertical in the background.
Yep, okay. It's fired.
And then also, since are we sitting down, Bob? Or are we hovering?
No, we're hovering.
Daniel has requested a sample of sediment here for grunge, and I don't know, do we have the scoop aboard? Is that how you want it, Daniel, or do you want a core? I mean, it's pretty rocky here, so I can imagine the core is going to... Let's see what if he comments here.
I mean, right here it's rocky. There was a little bit of sediment off to where we were just at, to the left.
Do you want me to open the bio box, see if there's a scoop in there?
There is a scoop in there. I put it in there.
Okay.
Yeah, this would definitely have to be a scoop.
Well, we can try over here. You know, I don't know.
Yeah, this is good here.
I want a tube core.
Yeah, we have the scoop.
You want a scoop or a core? You want to try a core?
We can try. I don't think it's going to go in far. I think a scoop is probably better for this type of sediment.
Yeah, scoop would be better.
Okay.
Agreed.
Scoop by.
Yeah, and Daniel doesn't have a preference, so I think we should just go for the scoop.
There's either an octopus or a very large acorn worm at the top left. Near the crabs approaching.
Yeah, I see that.
And Sam, I see what you mean about it. Looks like there is an octopus on
Big
that
shadow to the right. Is that your arm?
What's that? Big shadow.
Yeah.
Oops.
Oh, yep.
Yeah, there's an octopus in the distance there.
Mm-hmm.
out of here.
So we are collecting the scoop of sediment for Daniel Geiger at the Santa Barbara Museum of Natural History, and he's been looking in it for small animals, particularly shelled animals. And he just noted in the science chat that we did get a question earlier if we ever worry about missing new species that are tiny, that we can't really see easily with the camera, and that this is one way to look for them by collecting the sediments they may be living inside of. So this sample is going to go back and get, and they're going to look for all those little animals living in and amongst the sediments.
Can we zoom in somehow?
Pretty dense stuff.
Scoop is uncooperative.
I don't know how thick of a layer of sediment is here, and where there might be hard rock underneath. Steve, where do you want to put this sample?
It's probably just going to go back in the box where it came from, which was the starboard F compartment.
Thank you.
That was,
What did you just unearth there?
I don't know.
I know.
Something
I was
orange.
digging something up.
Huh.
Where are you
What
looking?
is that? Huh.
Can we zoom in some more?
Ah,
Big
Ah,
rock.
pretty
very
rock.
orange
Wow.
rock. That's pretty interesting.
Almost looks, wow, that was metamorphic. That's interesting.
How'd you find that out of?
Did you put, was that in the scoop first? You had that in the scoop, didn't you?
Did it come out of this?
I didn't put it in the scoop.
Got a plant.
You know, Nicole might
Do
want
you
that.
want it?
Looks kind of cool.
Yeah.
Yeah, I think we should take it.
Okay, zoom out.
I mean, it technically came up in the scoop, so.
Definitely different from the rocks around it. Goodness.
Well, if you can fit it in the scoop, I don't have to call it a new sample.
Challenge accepted.
Yes, I'm waiting to describe this as either a scoop of sediment or a scoop of sediment plus rock.
Yes, our onshore geologist guy is very perplexed by this as well.
Could you say that again, Kurt? I don't think I heard the whole thing.